Cells in your body are constantly dying and being born. How often they die and how fast they're born depends on what type of cell it is. Skin cells are pretty short-lived. They're constantly being rubbed off. Red blood cells live for about 3 to 4 months. That's why you have to wait that long to give blood again, so that the red blood cells you lost donating are replaced before you give blood again. The longest living cells in the body are nerve cells. There's some evidence that nerve cells can divide during your life. But for the most part, the nerve cells you're born with are all you've got until you die. So it's not the number of nerve cells in your brain that makes you as smart as you are, it's how many other nerve cells they can communicate with, literally making connections in your brain. So why is it that some cells divide and others don't? For the most part, it has to do with how fast they grow. The faster they grow, the more often they must divide. Skin cells have to grow fast in order to replace the ones that are damaged and lost, just in the normal activities of everyday life. They're only so large that a cell can get before it's inefficient at doing its job. There's two reasons it becomes inefficient to have large cells. First of all, it's hard to move nutrients and waste in and out of large cells, and secondly, it puts a lot of demand on the DNA, which contains all the instructions inside the cell. Inside the nucleus of a cell is the DNA that codes for all the instructions that tells a cell what to do. The DNA of every cell in your body is identical. It's just that the white blood cell uses the instructions that tell it to make proteins that break down foreign invaders. Salivary gland cells use the instructions that make proteins that begin the process of breaking down food. And nerve cells use the part of the instructions that tell it how to make chemical messengers and send signals to other nerve cells. However, each of them has all of the others' instructions. So in a salivary gland cell, which makes a lot of enzymes that break down starch, that section of the DNA gets used very frequently. So to be able to make lots of saliva with lots of enzymes, there need to be lots of cells that can each produce that enzyme, rather than having just one cell to make the enzyme. It's like taking a book out of a library. Imagine the library represents all the DNA in a cell, and each book in the library is like the instructions to make one protein. Let's say you want to read the new Veronica Roth book, Elysian. Because it's a really popular book, you might have to wait a long time to read it. If you didn't want to wait so long, there needs to be more libraries, or in the analogy of cell growth, more cells. Having more copies of the book at one library isn't an option for a cell. Each cell only has one copy of all of the DNA. The other problem with having large cells is that the cell produces too much waste to get rid of efficiently and requires too many nutrients to transport efficiently. How fast a cell can exchange material with its environment depends on its surface area. The more cell membrane it has, the faster it can move things through the cell membrane. If the school was on fire, imagine how long it would take to get people out if there was just one door. It's the same idea with having a small surface area. There are essentially fewer doors for substances to move in and out of the cell through. However, the rate at which substances in the cell are produced depends on how large the cell's volume is. Imagine a large cell that has 20 mitochondria, each breaking down sugar and producing carbon dioxide. Hopefully that cell has a large surface area to let all that carbon dioxide out, so it can get to the blood quickly and then out through the lungs. If not, carbon dioxide is going to start to build up inside that cell. It makes the inside of the cell acidic, and it will eventually start to affect the chemical reactions that should be taking place inside the cell. Remember, enzymes only work at certain small pH ranges. So the best scenario is that the cell has a small volume so that it produces a small amount of waste and has a large surface area to transport that waste out. When the surface area to volume ratio of the cell gets too low, the cell will divide, producing two daughter cells. Each of those daughter cells can function so that each of the daughter cells can function properly. The original parent cell must copy its DNA so that each of the two daughter cells gets one full set of DNA. The life cycle of a cell, or the cell cycle, is divided into two parts, the part where it's dividing and the part where it's not. When the cell is not dividing, it's in interphase. During this time, it's preparing to divide and carrying out its normal cell functions. During interphase, the cell grows and is producing new molecules. A cell that is dividing is in mitosis. Interphase is the period of growth between cell divisions. It can be divided into 3 phases based on the major events that are taking place. After a cell finishes dividing, it enters a phase called G1, which stands for gap 1. This is the longest phase where the cell is growing and carrying out its normal functions. The S phase comes next, and cells will only enter this phase if they'll divide. So most nerve cells never go through the S phase. S stands for synthesis, which means to make. So during this phase, DNA for a new cell is synthesized. Finally, gap 2, or G2, takes place just before the cell divides. Gap 1 is the longest phase of the cell cycle. This is where the cell gets larger and makes new proteins and organelles. It gets larger by taking in materials from its environment, breaking them down to create new proteins and other substances that it uses. If a cell, such as a nerve cell, is not going to divide again during its life, it will enter a phase called G0. This is a resting phase. Essentially, it keeps the cell from proceeding to the next phase, synthesis, where it would use a large amount of energy to make a copy of all of its DNA. After G1 is the S phase. It's during this phase that all the DNA gets copied. It's important that an exact copy is made so that the new daughter cells each contain a complete set of instructions. Also during the S phase, the proteins, like histones that are part of chromosomes, are made. During G2, that's the last phase before the cell divides. During this phase, the cell prepares to divide. Organelles like the centrioles are assembled, which will assist the cell in division. DNA is also proofread during this stage to make sure there's no mistakes. Some mistakes can be repaired, but serious mistakes or a large number of mistakes could mean that the cell will receive instructions to undergo programmed cell death. This essentially means that the cell kills itself in order to avoid creating problems in the body. A cell with incorrect instructions could cause cancer where it will divide out of control. Go back and use your notes to make a timeline of the cell cycle or the life cycle of a cell.